Okay, good afternoon and welcome back to the Business First Stage presented by IFT's Food Technology Magazine and Omnivore Podcast. It's great to see you all here. I'm Bill McDowell, the Editor-in-Chief of Food Technology and Vice President of Content Strategy at IFT. This afternoon, we're going to be exploring ultrafine bubbles, which is an emerging technology that's generating a lot of interest in the food and beverage space for its ability to optimize fermentation, preservation, and even sustainability. Today's presentation, how ultrafine bubbles can improve product quality and operational performance in food, beverage, and fermentation applications, sponsored by Hydrosome Labs. We'll explore how suppliers, manufacturers, and brands can leverage ultrafine bubbles to transform traditional practices and address contemporary challenges in food and beverage production. We're going to save some time for Q&A at the end, so hold on to your questions and we'll get around to you. But without further ado, I'm thrilled to turn the stage over to Dr. Nick Jackowitz, who's Principal Scientist at Hydrosome Labs, who's going to present on this fascinating topic. Nick?
Thanks so much. Appreciate it. Thanks for having me. Pleasure to be here today to talk to you about this really interesting topic. As we mentioned, ultrafine bubbles is an area that is very evolving and is a new area of science that we're looking at. And happy to talk to you all today and give you some of the ground sort of education on this exciting new area. So just a bit of background on myself. Again, I'm Nick Jackowitz, Principal Scientist of Hydrosome Labs. My background is from Cornell University, studied food science there and food fermentations, including wine chemistry. When I finished up at Constellation, I worked for Constellation Brands for a number of years, looking at product development, formulation, but now really getting into this new space of nanotechnology and these new types of sciences around ultrafine bubbles in that area. So excited to talk to you about that today. For thinking about, Thinking about ultrafine bubbles, it's really new. We have a technology to be able to create these ultrafine bubbles, a patented technology that uses force and pressure to flow liquids through finely engineered openings and devices to create ultrafine bubbles with a certain size and concentration. And what that does at the end of the day is we're able to increase what's called mass transfer, so we're able to move gases and solutes very effectively using these little bubbles. Imagine there's lots of them in solution, hundreds of millions or billions of them in solution. They can have tremendous surface area and move things very effectively, both solutes and gases. So we'll talk a bit more about that today as well. This technology, as I mentioned, is incredibly new. If you look back at the origins of ultrafine bubbles, it starts in the 80s in Japan, where they first noticed them in the ocean. They found them in crashing waves. And since then, we've seen the peer-reviewed literature really take off in terms of what ultrafine bubbles can do in their application areas. So we're seeing tremendous growth in the past 10 or 15 years, really looking at the fundamentals of this technology in application areas. Only in 2017 did ISO actually put some standards around what an ultrafine bubble really was, defining it as a bubble that's less than 1,000 nanometers or 1 micron. In practice, though, most of these ultrafine bubbles are typically 100 nanometers or less, so they're very, very small. And it's typically an area that was looked at was wastewater treatment in agriculture in the past with this technology. At Hydrosome, we're really bringing this into new categories like CPG, life sciences, fermentation, and we'll talk more about that today as well. It's a really exciting, fascinating topic. Some basic principles to get out of the way first when we think about ultrafine bubbles. They're not like classic bubbles that we see every day. They're not like bubbles that we would see in our beer or champagne or sparkling water that float to the surface. These bubbles are so small, they're actually not buoyant, so they don't float at all. So imagine you can have bubbles in solution that stay in solution for quite some time. So that's an interesting principle of them. They also, because there's so many, they have tremendous surface area. So their ability to interact chemically and biologically is quite massive. And that's a very important principle of their how they are used in different applications. The bubbles themselves are really just bubbles. So we're not adding any surfactants. We're not adding any chemicals to the equation here. We're making bubbles through force and pressure. The bubbles on the surface and the transition between the gas and the liquid are something called hydroxide ions that are naturally present in water already. And so those cluster around that interface, and that's what holds that bubble together over time. And so nothing else is added there. An interesting phenomenon when we talk about nanochemistry and we get really small is the properties of pressure that can be in these bubbles. So as we shrink a bubble down smaller, something called the Laplace pressure, which is due to the curvature of that bubble, really increases. So for example, a bubble that has a diameter of 100 nanometers, which is very similar to what we're making with our product, has a theoretical pressure of 31 atmospheres. So the ability of these bubbles to carry gas and be like a little gas tank that can hook up to a cell is quite impressive. And so we're still learning more about that, and we're pushing those boundaries there too, but the gas carrying capacity is quite high. And then lastly, this is sustainable technology. We're thinking about these nanobubbles. Again, no added chemicals, nothing else added here, and very low energy needed to actually make them. So it's pretty fascinating. If we're thinking about these bubbles as well, some of the unique properties that we bring to the table is stability. We think about a bubble that we see in our champagne or in our Coca-Cola. Those bubbles are transient and they go away quite quickly. However, these ultrafine bubbles can last for up to 1 year in a liquid and up to 2 years in a cream or formulated topical product, so quite some time. They're very stable, and that opens them up for use in new application areas like CPG. They have a very unique size that we're looking at as well. I mentioned the bubbles being less than 100 nanometers. We work with universities looking at using electron microscopes to actually quantify these, and we've seen bubble sizes less than 50 nanometers, some of the smallest that have ever been observed in the peer-reviewed literature. So very, very tiny bubbles. And we really pride ourselves on customization. So think about different concentrations that we can make, but also think about a bubble that's filled with different gases. I think we think about bubbles filled with air, and that's the bubbles we typically encounter every day, or say carbon dioxide, CO2. But what if you could fill a bubble with pure oxygen, pure hydrogen, pure nitrous oxide? What would that do? What would that do for fermentation? How could that affect taste and mouthfeel and organoleptics? That's an area of active research we're looking at right now. Again, because these bubbles don't float, they can stay in solution for quite some time. How can that affect sensory? I think that's a really exciting area of scientific research right now. And sustainability, obviously, too, is a big one. Clean label, green technology. I think there's a lot of scientists in the room, too. You want to know how this works and how these bubbles actually do their thing. And so we describe it by a concept that we call attract and deliver, where these bubbles act as aggregators. They're slightly charged because of that hydroxide ion surface. They can aggregate different solutes that are in solution and bring them in close proximity to the bubble, and they can deliver that very effectively to a cell, so the cell has better access to it. Again, remember the bubble is filled with gases and air, so they can also transfer gases more effectively too. So in an aerobic system, if you're trying to feed oxygen, say, to a cell, this is an effective way to do it, and that's the concept. To put this into practice, we have a test that we call the M&M test, which articulates this attract and deliver quite nicely. And so here you have 2 beakers filled with M&Ms and the same amount of water. Let's see. Yep, here we go. And the one on the top has ultrafine bubbles in it. That's a hydrosome water. The one on the bottom is deionized water. Same water chemistry, same temperature, just the influence of ultrafine bubbles. And you can see the diffusion differences between these 2 beakers. We haven't changed anything else. It's just the presence of bubbles. And again, it's their ability to attract those different solutes, in this case the colorants, to the surface and move them through solution faster than diffusion alone. And so that's the concept we're thinking about when we're talking about, say, a fermentation, moving things faster, or other application areas too. Just really having these bubbles transfer mass, transfer gases very effectively. This area of ultrafine bubble research is, as I said, is very new, and so there's limitless possibilities really where we can take this. We're hyper-focused on CPG and fermentation at the moment, but there's lots of application areas in agriculture, wastewater treatment, and some now emerging in the life sciences as well. So when we're thinking about ultrafine bubbles, where we can use them, we really think about things like, am I trying to enhance delivery of active ingredients to a cell or to a person? Am I trying to increase the efficiency of a process, use less time, use less energy, things like that? Am I trying to have higher yields? These bubbles can help in that function as well. And am I trying to keep things sustainable and have a clean label at the end of the day? Those are things to keep in mind as we think about application areas for these ultrafine bubbles. We're going to go through some different areas of application, looking at ag, we're going to look at fermentation, we're going to look at beverage. So we're going to start in agriculture. This is where it classically started. One of the best examples looking at the use of ultrafine bubbles in ag is in germination. So simply using this water with these bubbles to soak seeds enhances the germination rate. So we see faster germination rates and yields over time. So here in this example on the left, you have deionized water that's used to germinate these radish seeds. And on the right, we have the hydrosome. 7-day period, same time, same exposure. You see the vigor, you see how much more growth we have in those radish seeds just with having those ultrafine bubbles present, right? So that's a little little tweak we can make, but a big consequence, big difference there on the outcome, and that's well known sort of phenomena of ultrafine bubbles. We've done some work as well in the cannabis space, looking at how we can improve the growth of cannabis plants. And here we did a propagation study with the University, North Carolina State University, looking at how we could increase the root mass of these propagated pieces of cannabis. So they're all cannabis propagations off of a mother plant, and we looked after 13 days how the growth of these root systems evolved over time, and we noticed that every time the hydrosome roots were stronger, bigger, and had greater root mass. So this bodes well for them in the future as they continue to grow. We further that up with some work with a cannabis grower in Oklahoma, looking at increasing yields. This is what it's all about at the end of the day, is can we make more cannabinoids, THC, CBD? Can we make more terpenes, which are really responsible for the positive notes and why people like certain cannabis strains? And we noticed when we supplemented with the water, we got better yields of those cannabinoids and better yields of terpenes too, quite dramatic, in fact, on the terpenes, over 50% increase. So just by making a small swap, we got that benefit. And actually, we added less fertilizer to these as well and still had these benefits. So it's cost savings as well as yield improvement. On the food and beverage side, there's some really interesting applications, still very new in food and beverage space, I'll say, in terms of ultrafine bubble use. It's mostly, again, in ag and wastewater treatment. We've done some work with The Ohio State University, looking at sensory studies and understanding how a beverage or even a water infused with ultrafine bubbles, what does that taste like? How are people perceiving that? And one common sensory note we got back was cooling. People found that the water had a cooling effect with no other chemicals, no other additives added to it. They just noticed that that was on the palate. It was more cooling. And so we did some work with Ohio State. We found that there was a directional difference between the samples, but not statistical. So we said, okay, there's something here. There's something going on with these bubbles. We can perceive them, but let's take it a little bit further and look at something else. So our next thought was, can we infuse these bubbles with something other than air, something that's not as inert, and how does that taste? So we did some work with a gas called nitrous oxide, which is commonly used even in whipped cream canisters and in that kind of application. But instead of infusing whipped cream, we were infusing bubbles. And what we noticed was when we gave people a triangle test and presented two samples, both infused with nitrous oxide, one with ultrafine bubbles and one without, we noticed there was very big differences in terms of the perception of sweetness just with these ultrafine bubbles being present. And that was pretty impressive and pretty, we were surprised by that, and we're doing further research to really play with this, tweak the concentration of these bubbles, and really push this further, because how does this sweetness impact, again, a beverage? Does this mean you have to add less sweetener? How does this change other flavors? How does this change mouthfeel? And then the next question, obviously, is what about other gases? What do we think about that's nitrous oxide and that's air? What about pure oxygen? What about pure nitrogen? We've all maybe tried or heard of Guinness, where you have those small bubbles that obviously impact mouthfeel. What if you had a bubble that was 100,000 times smaller than a Guinness bubble? What does that taste like? How does that affect organoleptics? I think that's something we're really interested in, and working with a lot of beverage partners right now to understand how we could really bring this forward. So looking at all kinds of gases, including even CO2. What does a nanobubble filled with CO2 taste like? How does that impact things? So lots of research in this space, and so we're really excited to collaborate in this field more. Kind of switching gears, though, further, we are doing some work even with ultrafine bubbles, looking at how they affect humans and how they affect the microbiome, the gut microbiome. There's some research that's been shown already that the presence of ultrafine bubbles in drinking water can affect the diversity of the gut microbiome with no other changes to diet. So that's been observed in some mice and lab rat studies over time, and we've seen those positive changes, both when it comes to that microbiome diversity, even short-chain fatty acids. And this is without having to do probiotics, prebiotics, just looking at supplementation of water and changing to the gut. So we took this and said, okay, we'd like to use our water in this case and really look more at a preclinical study using lab rats where for 12 weeks, one group only gets ultrafine bubble water and one group only gets regular deionized water with some minerals added back. And let's look at the differences between their gut microbiomes. Let's look at other health markers like inflammatory markers, liver enzymes, blood glucose levels. How does this all affect mammals in this case? What are the implications for humans? I think we're really excited to see how this study turns out. It's almost finished right now. It'll conclude the end of this month. So a lot of really interesting possibilities when it comes to mammalian research as well. And then finally, I think a really interesting area that we're pushing into very diligently at the moment is fermentation. So again, if we have these bubbles, they're able to provide mass transfer of gases and solutes. How can that affect the yeast cell, E. coli cell, filamentous fungi, mammalian cell? How do these bubbles help in those processes? So at Hydrosome, we're looking really at an area of fermentation that's often overlooked. So if you think about folks that do a lot of precision fermentation, there's been a lot of money invested in cell engineering, adapting E. coli or yeast cells to pump out a certain analyte of interest and do genetic engineering on them. Conversely, there's been a lot of downstream processing work. How do I extract what I've made more effectively? How do I get that better yield? But no one's really worked at the fermenter itself and really looked at that and how can I improve upon that fermentation process. Typically, they'll add an aeration stone or some agitation, and that hasn't changed in the past 2-3 decades. There's a lot more we can do in this space, and I think these ultrafine bubbles really play an important role to unlock this area that we're looking into. So that's where we focus. If we think about how these bubbles can be used, we can use them a couple different ways. We can think about, in a fermentation, using them in a batch process. So let's infuse some water. Let's add nutrients to those bubbles in that water, and then let's sterilize, and then let's inoculate, and we go from there. Conversely, if we have a fermentation where the bubbles will be consumed, say in an aerobic fermentation, aerobic process, we do what's called a continuous process, where we actually add bubbles continuously so that if they are used up, if that gas is consumed, we can add more back in. And so we can have almost like a modified pump over during that process, during that fermentation, and we can have those bubbles added in continuously. So when we looked at batch processes, we've seen differences, we've seen benefits, especially on the yeast and yeast side. So here's some data looking at Pichia pastoris, which is a common cell line used in precision fermentation. We're seeing boosts in overall biomass there. This is with a partner in Vancouver, Canada, that was doing some work with Pichia. And then we've done some work in the lab as well, looking at Saccharomyces cerevisiae, just seeing better growth, shorter lag phase, stronger exponential phase growth, just with a batch process. We also have collaborations with folks that do filamentous fungi research, looking at how can we produce alternative proteins this way as well, using the biomass from filamentous fungi. And they too saw differences when using these ultrafine bubbles in a batch process. So there's definitely something there we can move the needle, but definitely with these aerobic processes, we think by infusing more bubbles, we're getting more benefit. And then that's where we ended up working with the University of Illinois IBRL lab down in Urbana-Champaign, looking at doing a E. coli fermentation where we constantly infuse ultrafine bubbles at a 400-liter scale and looked at the growth of those E. coli over time to see how they were affected. So we had 2 different treatments, one with bubbles, one without bubbles, and we wanted to see the growth parameters that occurred from those 2. What we noticed was a pretty marked difference in terms of the growth profiles, where with the ultrafine bubbles, we had a shorter lag phase, and that fermentation really took off after about 30 hours or so, and having a very fast doubling time, versus the control, which did not have any ultrafine bubbles in it, everything else being the same, and we noticed that it took over 40 hours to finish fermentation, so about 25% longer to do. So we're seeing efficiency gains using these ultrafine bubbles, and we also saw that the overall cell biomass was improved by about a factor of 2. And so definitely we're getting things to these cells, getting them what they want more effectively, and that was really positive in this case. We looked at some of the other parameters during that fermentation as well, and noticed things like the doubling rate was about 50 minutes using the Hydrosome ultrafine bubbles versus 100 minutes for the control, so about twice as fast. We also looked at how these fermentations converted glucose into biomass, and noticed it was about 3 times more effective changing that glucose into biomass. So again, glucose being a very expensive ingredient that you're using during your process, so we can convert that more effectively into cells, what we saw. We're doing a lot of work right now understanding more about metabolism when these bubbles are present. So we have RNA sequencing that's currently underway, looking at how these bubbles can affect metabolism overall. So we've seen differences in things like acetate and ethanol, which are sometimes markers for fermentative processes. So the fact that in the hydrosome treatment, acetate and ethanol are lower may mean they have better access to oxygen, which is, again, what we're providing in our bubbles as well, too. So we're looking more into that and how gas transfer plays into it, but some marked differences in metabolism and carbon flux and carbon flow through these cells. So at the end of the day, what we really saw was just a much shorter fermentation time, faster process overall, higher biomass, and again, in these types of situations, we're using or adding these bubbles into fermenters that are already present on site. So we're not adding new tanks or new infrastructure. We're using what's already there and really trying to upgrade that fermentation process by adding these ultrafine bubbles on the side. So that's very easy to implement, fully automated, and really easy to use. So it's there's a lot more to be seen in the fermentation space, and we're excited to keep exploring it. I guess to conclude, what we're really seeing is really doing a lot more education in this ultrafine bubble space. It's very new. Most people we talk to have not heard of it, but really the possibilities are quite limitless when it comes to this technology and what it can do. Again, we're seeing these different opportunities in agriculture classically. We're seeing things in industrial when it comes to doing fermentations, and also consumer packaged goods. There hasn't been a lot of innovation in the space when it comes to, say, beverages with ultrafine bubbles, or even food products as well. And we think there's a lot of opportunity to explore how these bubbles can be manipulated, concentrations and gas-wise, to positively affect these different foods and beverages. So we just think there's a lot of opportunity in this space. And again, it's a whole area where if you're looking at efficiencies, yields, and being cost-effective, that's where these bubbles can come in a very sustainable and green way. And so I guess with that, I really want to thank everybody for their time and attention, and I'll open it up to any questions you might have. Thank you.
Nick, thank you. That was absolutely fascinating. And, the applications that you talked about literally touch on almost every corner of food and beverage production. So we want to open up for questions, but I have a question up front.
Sure.
You said up front that this is a new technology, but how is it possible that, nobody knows about this?
Yeah. No, it's a great question. I think it is going back to the 80s or so, but our ability to measure and see ultrafine bubbles has really only come into focus in the last 10 or 15 years. And I think that's why folks haven't been able to really do a lot of research, because they are so small. Imagine trying to see something that's 100 or 50 nanometers. It's really hard to observe optically even. So you have to use electron microscopes or things that are very sophisticated. So only in the last little while has the technology really caught up to the space. And so now I think the floodgates have opened a little bit more, and you're going to see a lot more research happening on this space.
Other questions? Please raise your hand. Yep.
Hi, I'm just curious about how do you measure the lasting period of this state?
Yeah.
I'm guessing it's not going to go eternally. So whether if it's in a liquid form, is it going to last that state of the ultrafine bubble for a long time, or does it die out in a couple of minutes or so?
Great question. Yeah, so the stability we've seen really depends on the complexity of the beverage and with the chemistry of that beverage. But in pure water, we've seen those bubbles persist for up to 1 year. Because again, they are so small, they are not buoyant. And because they have a negative charge on the surface, that means these bubbles actually repel each other. So they're not going to want to come together and coalesce over time. They have what's called a negative zeta potential. So that's actually quite strong, and that repulses them against each other. So they do last for quite some time, and we've seen them stay, again, for upwards of 1 year.
Other question? Oh.
So, well then, if you consume, let's say, liquid with the ultrafine bubble, then it might affect your digestion. If it stays in that state in your stomach, then it's going to affect your diet.
Yeah, I think that's an active area that we're looking into with this microbiome study is really banking on them surviving and getting into the gut to be able to make a difference. And that's what we're seeing in peer-reviewed literature already, that's the case, that they can persist and make a difference. If they just were rupturing in the stomach or in the gut, you wouldn't see a difference downstream. So something's obviously happening there where they're making their way through. They're actually very, very hard to break. Even through autoclave cycles, they persist just fine. they're very, very hard to break. So we think that they will have no problem getting through the gut, and we're just really curious to understand about how they can positively impact human health over time.
Do we have a question over here? Yeah.
If you put your hydrosomes, say, under external pressure, and also have you, I guess, studied the effects of, say, a hydrosome process versus a blank, so to speak, in terms of what the pH is of the resulting hydrosome mixture?
Yep, that's a great question. So we've noticed very minimal changes in terms of the physical properties of water. We don't see much shift in pH or volume even, obviously. These are very, very small bubbles, so they don't take up a lot of space. Again, the main way we look at them is through electron microscopy or through optical methods like dynamic light scattering or nanoparticle tracking analysis. So it's very hard to detect them beyond those methods I just mentioned. Yeah.
So if we add these hydrosomes to our beverage, like orange juice, and then we pasteurize, sterilize them, they won't destroy?
Yes, we've seen they have tremendous stability. It's one of the hallmarks of these ultrafine bubbles is they're remarkably stable. So again, even trying to break them through heat, we put them through several autoclave cycles and they persist. And we put them through different pH pH ranges and different temperatures, and they still remain. So obviously they're not going to be stable everywhere, and the shelf life will be different depending on the beverage that they're going into, and you would do stability studies to verify that. But overall, they are actually quite stable.
Even for freezing, like for ice cream?
So for freezing, that's actually one way to break them is by a freeze process, because a hard freeze will disrupt the bubbles and they will break over that period. So a hard freeze thaw will disrupt them. Yes.
Okay, thank you.
Hey, Nick, interesting talk. I just had a question. It occurred to me that this technology could be used to prevent or remedy stuck fermentations. Just want to get your thought on that.
Yeah, it's super interesting. I think we're doing a lot of work in the fermentation space, and perhaps if a fermentation is stuck because of lack of oxygen, say, if they're needed that to start that fermentation, an alcoholic fermentation, that could be a way to go. But just a really moving mass and really starting to mix these tanks with these bubbles too is another way we can help encourage homogeneity in those tanks, especially if yeast cells have fallen down to the bottom. It can help mix and help move those nutrients around a bit better. Yeah.
I'm not sure if I missed that, but have you did any trial in high-fat formulations if it affects the oxidation, positive or negative?
Yeah, so we haven't done a ton of food formulations at this point, but I will say one of the hallmarks of these bubbles, again, is the remarkable stability. So they really don't let out any of the gases that are inside of them. They hold on to that gas very tightly. So we haven't noticed any hyperoxidation in products that we've made. We do a lot of work actually right now in the cosmetic space. And so there, obviously, those products can be very prone to oxidation as well. And we haven't seen excessive oxidation or any negative issues that way, again, because all the gas that's inside those bubbles stays inside the bubble. It doesn't bleed out. Otherwise, those bubbles would cease to exist. So we haven't seen issues at that point, at this stage.
So Nick, when you're looking at the range of F&B applications specifically, and you mentioned a broad range of them, what is the next frontier that you are the most excited about? Like what's the one that is the furthest along that you think is really going to be the big breakthrough for this?
Yeah, I think in food and beverage for us right now, we've seen a lot of interest from beverage companies looking at this technology and saying, again, how can I infuse these bubbles into a beverage, and how can I impart different gases that might change the organoleptics of a beverage, right? We're all used to the perception of a CO2 bubble done classically and dissolved into liquid. But again, if we were to make that bubble now, thousands of times, hundreds of thousands of times smaller and make that 100 nanometers, how does your body perceive that? What does that do to other flavors that are in solution too? And then again, what about other gases? How can they modulate flavor? I think I'm really excited about how a beverage can be modified that way, because again, the stability of these bubbles and then what other gases can bring. Our work with nitrous oxide, which is known again to drive sweetness, is really encouraging in that area. So we're excited to try other gases and look at how, with different combinations of ingredients, how these bubbles kind of change the game there.
Fantastic. Everyone, let's give a round of applause to Nick for a fascinating presentation. Want to acknowledge Hydrosome Labs is a bronze sponsor of this year's IFT First, so thank you very much for your support of the program. Be sure to go and stop by their booth, which is 4474G, to learn more about the technology and about the company.